So there's several areas that we're focusing on, and these are not unique for lymphomas. I think many groups treating different types of cancers having similar approaches. So most of us are looking for biomarkers to identify patients or to select patients for appropriate therapy, precision medicine, what people call it. And that's mainly now depending on genetic sequencing methods. So we're sequencing every single patient who comes through the door at Memorial with lymphoma, really free of charge, as long as they're willing to give a consent. And we're trying to pull data and try to identify biomarkers that can predict response to certain therapies. The second area we're focusing on is acknowledging that single agents are very effective or curative by itself. So combination strategies, and that's tricky because if you keep in mind that there's probably about 600 new agents available for either in clinical or preclinical testing for cancer, and if you want to do combinations of these 600 agents based on educated guess, it will take forever. So everybody's trying to figure out how can we identify doublets or triplets in more of a rationally designed mechanism-based combination movement. And we're actively involved in this, trying to identify mechanism-based combinations that we can prioritize and say this doublet is better than this doublet, therefore I want to invest the next 3 years testing this combination before. And that's a really big thing. It's ongoing in terms of identifying new combinations. And the third one is immunotherapy. Again, immunotherapy took everyone by surprise, especially knowing that these new agents, checkpoint inhibitors, active in diseases that are not typically responsive to immune therapies like lung cancer and colon cancer. So it's a big thing. And people think of immune therapy mainly as antibodies, but there's more platforms to immune therapy. CAR T cells, for example, a completely different platform, right? Bispecific antibodies are coming back with really vengeance with two-headed antibodies. And then the old-fashioned naked immune checkpoint inhibitors. And then you can think about combination of these different platforms and combination of these platforms with chemotherapy and small molecules, right? So we're actively involved in these kind of approaches, in immune therapy based with different combination strategies.